Welcome back, friends. In this video tutorial we'll be talking about Salmonella and mostly about the pathogenesis caused by Salmonella and the different virulence factors produced by Salmonella to cause the pathogenesis effect. Now, so that's why the term came here, the molecular aspects of Salmonella pathogenesis. We'll be majorly focusing on the molecular approaches and the genetic changes in Salmonella that are brought about huge changes about the infective property of Salmonella. So let's begin. An overview of the clinical syndrome of Salmonella is provided here. Normally, the clinical syndromes that Salmonella provides are two things. One is the enteric fever, which is caused by the enteric type of Salmonella, which is caused the typhoid here. And second thing is the gastroenteritis caused by a different variety of Salmonella. So the enteric fever here is caused by Salmonella typhi or Salmonella paratyphi, which is the infection due to normally the food and waterborne diseases. They are carried through water and contaminated food. And normally another one, which is gastroenteritis, is caused by Salmonella typhimurium or Salmonella enterica. Okay. So normally this enteric fever or typhoid that we have talked about, caused by Salmonella typhi or Salmonella paratyphi. The clinical manifestations are fever, abdominal pain, related with diarrhea, and maculopapular rash. These are all examples. And for the hallmark of enteric fever, it is mononuclear cell infiltration. That means mononuclear cell means cells containing only one nucleus. Those cells are kind of damaged. Then hypertrophy of reticuloendothelial cells and inflammation in the intestine, intestinal prayer patches in intestinal inflammation. Then spleen and bone marrow inflammation, and as a result, so normally without treatment the mortality rate is kind of 10 to 15% in Western countries, but in countries like third world countries like India and Africa and all those places, this mortality rate is a little bit higher. Now, normally there are many of the non-typhoidal Salmonella strains are there, among them Salmonella enteritidis and Salmonella typhimurium. These two things are important to cause the enteric, so gastroenteritis normally. Now, I remind you that many people think that Salmonella typhimurium, as the term says, typhi, they cause typhoid, but these are not the causative agent of typhoid. Salmonella typhi and Salmonella paratyphi are those which usually causes typhoid or enteric fever, but Salmonella typhimurium and Salmonella enteritidis, they causes gastroenteritis. Now, usually the cause is self-limited enteritis in human, normally intestinal inflammatory response as well as secretory response is caused due to this gastroenteritis, intestinal inflammation as well as secretory response, which is also called as flush diarrhea. Now, in certain strains like typhimurium, infection proceeds an illness resembling enteric fever that served as experimental model for systemic Salmonella infections, but in very few cases. That's why the Salmonella typhimurium is added there. An overview of the clinical syndromes. Again, if we look at here, all Salmonella which are typhoid or non-typhoidal infections begin with the ingestion of the organism in contaminated food or water that we've already talked about, and gastric acidity normally prevents a significant internal barrier on the first initial barrier against this kind of infection, because Salmonella exhibit very less acid tolerance, but they can have an adapted acid tolerance response exposure to lower pH conditions like the stomach acidity. Now, after entering into the small intestine by passing through the stomach and everything, surviving in the acidity, Salmonella can traverse the intestinal mucosal layer using its flagella. Because it's having the flagella, it can move and it can traverse that intestinal mucosal layer. As it is traversed through the intestinal mucosal layer, then it encounters and adheres to the cell of intestinal epithelial. And the addition of the Salmonella with the intestinal epithelial is brought about by the cellular extension of their body, which is called fimbriae, to adhere with them. And then, so you can see here in this picture, using flagella, the Salmonella bacteria can easily make their entry through the mucus layer. So this is the mucus layer they are making their entry through, and this is a much more higher resolution structure in electron microscopy. You can see the smaller fimbriae outside. Flagella is helping them to migrate and propel through this barrier, and fimbriae is helping them to adhere to the epithelial cells. Now, Salmonella invade the epithelial cells by morphologically distinct processes called the bacterial mediated endocytosis or directed uptake process. So what is directed uptake? When the bacteria goes and attaches with the intestinal cell or epithelial cell, it provides some signaling molecule to the epithelial cells and it modifies the epithelial cell surface in such a way so that it can form bulge or vesicle-like structure within which it can entrap the bacteria. And that formation of vesicle is mediated due to the membrane modification change called membrane raffling. And this membrane raffling is brought about due to the modification in the cytoskeleton or actin cytoskeleton that is present inside the cell. So it modifies the actin cytoskeleton of the host epithelial cells. As a result of that, it kind of changes its structure, it kind of engulf that bacteria inside, and then the bacteria is inside the vesicle, it will be transferred, internalized inside the cell. This process of membrane modification is called membrane raffling, which keep on changing and uptaking bacteria inside, and that is mediated by the release of some factors by bacteria. For example, here this is, you can see the HEP2 cell is the host cell where the bacteria is kind of internalized. As you can see in this picture, these green colored sections are the host cell membrane, which kinds of form like raffles, like cabbage, and it kinds of bacteria kinds of make entry through these points. Now, this bacterial mediated endocytosis involves profound cytoskeleton rearrangement that I have already talked about, and it requires coordinated synthesis of multiple bacterial proteins. That is very, very important. Now, there are specific host signal transduction pathways that govern the cytoskeleton organization, and to trigger those signal transduction pathways, they need to secrete some important protein mediators. Now, once they enter inside the cell, as you can see here, this is the Salmonella bacteria that come here and in contact, and then internalization occurs. Once the internalization occurs in the epithelial cells, these are the epithelial cells marked at E, and this is a cell called M cells. These are also part of the epithelial cells but plays a vital role in sampling antigens, because these are antigen sampling cells which pick up some antigen and drag it to nearby neutrophils so that neutrophil can take care of them and our immune system can take care of them. Now they are having a very important strategy to bypass these M cells. This is also very important. To bypass the host immune system, they are escaping the function of M cells, and they are only making their entry through the normal epithelial cells. So as they are making their entry through the epithelial cells, they affect three different ways. One thing is that after entering into this cell, what they cause the cell to release massive amount of water and chloride ions outside due to the ATP ribosylation inside, a small kind of modification inside the cell. It triggers the water and chlorine ions to be released outside. So it causes the diarrhea for that reason, and this epithelial cells kind of dehydrated. The second way of response they are providing is as they are present, they are having different antigens in their surface. They are having their membrane, their capsules, and every round of structures which will trigger the synthesis of interleukin 8 by these epithelial cells. And this interleukin 8 is a chemical mediator that will bring other type of immune system cells like neutrophil, macrophages, and other cells into this scene. So that is the second inflammatory response that is caused. That's what we have talked about this gastroenteritis. There are two important causes. One is it will bring upon the inflammatory response, and second thing, it will also bring upon the watery diarrhea. And first thing is established here, inflammation is established, and the diarrhea we have already talked about how it is established. And the inflammation is further caused due to the release of this IL-8. Neutrophil are kind of traversed through now through this epithelial cell linings, and they will come into the intestinal lumen. So you can see the lumen is now filled with water, chloride ions, and it is filled with neutrophils and other molecules. Blood also can sometimes come through these places due to the internal epithelial damages. So watery diarrhea and bloody diarrhea may cause due to this activity of Salmonella. Now if we look at the process happening in the enteric fever system, so in enteric fever, again the entry of, or the route of entry for Salmonella causing enteric fever as well as causing the gastroenteritis are kind of same, but the approach is slightly different. So what they are doing in enteric fever, they are entering inside these epithelial cells, except for this epithelial cells, they are taking entry through the M cells. Remind you, M cells, we have talked before that M cells are the antigen sampling cells found in the epithelial lining in the of our intestine. So this M cells will take up the bacteria, it will chop them up, it will process them, and it will provide them to the nearby immune system antigen presenting cells like macrophage, neutrophils and other cells. So here they are engulfed by these M cells, and they are processed, and the M cell will process them and finally provide them to the macrophages, which are very dangerous player of the immune system of host. So the macrophage will uptake them using toll-like receptor here, by the toll-like receptor mediated endocytosis inside, and then inside them it will chop them up and do the rest of the job. But what they are doing here, the Salmonella are entering inside the macrophage by their choice. They want to be entered inside the macrophage, because inside here the bacteria, this bacteria can stay inside this vesicle, inside this endosomes for longer period of time, so that they can be carried through the bloodstream by macrophages. So the Salmonella in this case using this macrophage as their transport vehicle to set, because they want to move and migrate through different section of the body, and that can only be achieved if they are entered inside this macrophage, and they are achieving that task beautifully. And once they are inside the macrophages, they start to secrete tumor necrosis factor and other factors that will bring upon many other immune system cells, but they are keep on living inside the macrophage, and they are carried from one place to another place via the bloodstream. Now in any kind of infection, the actual goal of the bacteria is to reach the blood vessel or the blood system, because if they reach the blood system or the circulation system, it can migrate from one place to another place very rapidly. Now the Salmonella virulence mechanisms are mediated by pathogenicity islands, and that's a very interesting concept. Now pathogenicity islands are a stretch of multiple functionally related genes necessary for a specific virulence phenotype. They are called the pathogenicity islands. So multiple functionally related genes placed one after another in a stretch of genome sequence, they are specific for a particular virulence phenotype. Okay, and they are acquired normally via the horizontal gene transfer machinery. So if you look at here in the Salmonella pathogenicity island, they are having two important pathogenicity islands. One is Salmonella pathogenicity island 1 or SPI1, another one is Salmonella pathogenicity island 2 or SPI2. If you look at here in the Salmonella pathogenicity island 1, you can see a lot of different genes are present there. I won't encourage you to memorize the names, you don't need to memorize anything about that, but try to look at it, and what you can find here that there are different sections, different genes causing different proteins, SIC protein, SIP protein, SIP, SOP, SPA proteins, INV proteins, and many different varieties of proteins. Try to just understand that these proteins are important for some of them are important for degrading the host cell, some of them are important to bypass the host's immunity mechanisms and many things. And all of these genes, most of these genes that are embedded here, they are arised due to the horizontal gene transfer among the different bacterial species. Now if we look at the pathogenic island 1 in detail, what we can find, it encodes the gene necessary for invasion of intestinal epithelial cells and induction of intestinal secretory and inflammatory response. That's what the SPI1 is doing, invasion of the intestinal epithelial cells and induction of secretory and inflammatory responses. Now the Salmonella pathogenicity island 2 genes encode the intracellular survival and replication, and also the establishment of systemic infection beyond the intestinal epithelial cells. So if we look at here, SPI1 is very much important for those Salmonella strains that are causing gastroenteritis, which are causing the inflammatory as well as the flush diarrhea. On the other hand, Salmonella pathogenicity island 2 are required for those strains of Salmonella which usually causes the enteric fever, like Salmonella typhi. Now both these SPI1 and SPI2 encode specialized devices for the delivery of virulence proteins into the host cell. This is termed as the type 3 secretion system or TTSS. It's a very interesting way of secreting these protein molecules from this bacteria to inside the host cell like a syringe of injection. So if we look at the type 3 secretion system, it is simply manipulating the host cell with kind of molecular syringe. So you can look at here, this is the Salmonella, this is the inner and the outer membrane, and this membrane there are protein shafts that is building the basement of the type 3 secretion apparatus, and from this basement a needle like hollow chambers start to form and assemble, and this hollow needle will go and it can penetrate the infected host human cells. Now as it is penetrating these infected human cells here, so what they can do, the Salmonella will produce some protein mediators or factors. Those mediators can easily now pass through this type 3 secretion apparatus needle and can enter into this infected human cells via a small channel produced in the cell membrane of human cell. It is called the translocon. Now through the translocon, those type 3 secretion apparatus spread their protein molecules in the infected human cell, and that's how those protein will work there, and they are going to work and they will hijack the host cell immune system machinery and everything like that. Okay, so it kinds of breaches between the bacterium and the host during the secretion of effector proteins, which are required to create the active level of infection in host organisms. Now the virulence proteins translocated into the epithelial cell by SPI1 TTSS, that means type 3 secretion system processes. Now two factors or two effector proteins among them called SOC-E and SPTP that targets the membrane of Rho family or Rho family or GTPase protein or the G proteins, right? So the members of G family including the CDC42, RAC, Rho, these are all example of the G protein. So this SOC-E and SPTP targets these G family proteins, and they are kind of regulate the gene expression of these G family proteins, and through the members of the mitogen activated protein kinase family or MAP kinases families, using the MAP kinase proteins downstream, they are controlling the transcriptional regulation of those G proteins like CDC42, RAC and Rho, which plays a very vital role during the virulence. Except for that, they are also secreting some effector molecules like CYP-A and CYP-C, which interacts directly with the host cell actin cytoskeleton, and it modifies the host cell actin cytoskeleton. And why this modification is required? Because they are changing the host cell to take them inside. We know that they are going through that directed uptake process. That's the actual goal of Salmonella entering inside the host cell, the directed uptake by modifying the actin cytoskeleton, and they're achieving that task using the type 3 secretion system secreting CYP-A and CYP-C proteins. And then except for that, another effector protein called SOC-B promotes the intestinal inflammation and fluid secretion by subverting the inositol phosphate signaling pathway. You don't need to go through the detail, but just know that there are majorly 3 different types of effector molecules. Previously we have talked about SOC-E, that is responsible for controlling the transcription of the G proteins like CDC42, RAC and Rho using the MAP kinase molecules. Then the second type are CYP-A and CYP-C, which are important for modifying the host cell actin cytoskeleton by directly binding to them. And the third kind are SOC-B that promotes the intestinal inflammation and fluid loss. And if you look at the another important approach, except for the type 3 secretion system by this Salmonella pathogenicity, and that is 2 component response regulators called the 4P and 4Q system. Now they consist of the membrane spanning sensor region and obviously kinase protein that is doing the downstreaming part. Now here the kinase protein is PHOQ or 4Q that phosphorylates the second cytoplasmic component, which is 4P. So 4PQ component, 4Q is the sensor, 4P is the downstream regulatory molecule. So 4Q senses the environment using the environmental stimuli, and that will transfer the signal to the cytoplasmic component. Here it is 4P, and then the 4P will phosphorylate certain down regulatory molecules, and finally the cytoplasmic response will occur, and finally the transcriptional regulation of certain important genes inside the host cell will occur. So 4P activated genes are expressed within macrophage phagosome and that are required for the survival of that bacteria of Salmonella inside the macrophage phagosome. Remember, this particular 4P 4Q system is not required for those strains which are causing gastroenteritis. These are only required for those strains which are causing the enteric fever or typhoid, because in this case they want to stay inside the macrophage. For that reason they need to regulate some important protein mediators. They need to produce some important proteins or chemicals which will help them to stay and be living inside those vesicles or endosome vesicle in macrophage, and that is mediated by these 4P activated genes. And 4P activated genes also promote the resistance of antimicrobial peptides by catalyzing some covalent modifications of the lipid A component of the lipopolysaccharide or LPS layer that we are going to see. Now we have talked about the Salmonella pathogenicity island 1 type of proteins. We've seen that this SP1 proteins are producing some mediators that are going to act via the type C secretion system processes, and it is producing SOP-E, SOP-C, CYP-A, CYP-C and all these molecules which are required for causing enteric fever as well as gastroenteritis. We have also seen a 4P 4Q mediated sensor kinase applications that are also regulating the enteric fever disease. Now we are going to see the virulence of proteins translocated in epithelial cells by the SPI2. Now most of the time we know that SPI2 are producing some important factors which are required only for those strains combining the long run, that is those cells which are causing enteric fever like Salmonella, typhi or paratyphi. So usually these proteins translocated into the epithelial cells, and this system activates within the phagosome and translocates bacterial effector proteins from the phagosome into the macrophage cytosol, because that is the region they require this kind of proteins. Now the effector proteins they produce, it will interfere with the maturation of the Salmonella containing phagosome and thus preventing the phagosome and lysosome fusion. Now that is a very and probably the most important part for the Salmonella to survive inside the phagocyte or macrophage, because it needs to block the phagolysosome fusion, otherwise the bacteria will die due to the acid stress, and they are deliberately doing that using SPI2 mechanisms here. Now SPI2 is also relying on the type C secretion systems, but it is connecting only between a channel of the endosome that inside which the Salmonella is present. Now this SPI2 prevents co-localization of Salmonella containing vacuoles with the NADPH dependent oxidase that catalyzes the production of reactive oxygen species within the phagosomes. So they are modifying many things, they are producing these reactive oxygen species that are damaging things and everything like that. So if you look at here the virulence factors involved in the macrophage survival for the systemic infection by Salmonella, that is the systemic fever or typhoid fever. So if you look at here, Salmonella binds to TLR2 or TLR4 in dark inside. Now in this section they start to secrete some important mediators which will block. So you can see using SPI2 effector molecules via the type C secretion system, they are releasing some chemical mediators outside. They are blocking the phagosome lysosome fusion, as well as they are helping the bacteria to grow and divide inside the vacuole. That is very important. So they are not only being uptaken inside, they are not only secreting factors, but they are also capable of dividing and keep dividing themselves inside the vacuoles inside the macrophage. Okay, and they are having a beautiful mechanism of preventing the oxidative stress created by the macrophage to kill them using nitric oxide mediators, because nitric oxide found to be most dangerous of the reactive oxygen species to kill Salmonella. It is producing several kind of other enzymes like superoxide dismutase or hydrogen peroxidase to break down some such kind of reactive oxygen species, but nitric oxide is that much dangerous, it won't restrict it using certain kind of enzymes. So to restrict it using some important task is required, and the task is fulfilled by Salmonella using Salmonella pathogenicity island 2. So if we look at here in this picture, it is telling us the important fact. So this is the Salmonella cell, this is the phagosome, and what Salmonella is doing here, Salmonella is secreting some important SPI2 mediated proteins outside that are required, right? The effector molecules, so effector proteins. Here such effector protein is SPIC. They can produce and secrete outside via the type 3 secretion system. Okay, now normally what we know that inside the phagosome there is a lot of nitric oxide produced. Now this nitric oxide can easily go and can block or prevent this SPI2 or Salmonella pathogenicity island gene regulation or expression. As it is blocking the gene expression of Salmonella pathogenicity island 2, no further Salmonella pathogenicity island 2 proteins can be produced or can be translated there. So that is the job of nitric oxide. So somehow to survive inside the cell, Salmonella needs to regulate the concentration of nitric oxide inside the phagosome. So how they are doing that? Now we know that this nitric oxide is dangerous for Salmonella, but nitrogen dioxide or NO2 or nitrite, actually not dioxide, it's nitrite. This nitrite is not that much harmful. So if it converts nitric oxide into nitrite, this nitrite can stay there for longer run, and Salmonella can also leave, and it can also express the SPI2 mediated proteins. So what they are doing here, there is a balance between nitrite and nitric oxide inside this phagosome. So they are changing this balance. How? They are driving this nitric oxide out from the phagosome inside their cell. So as they are dragging this nitric oxide inside their cell, they are taking this equilibrium at this left hand side. As a result of that, as the amount of nitric oxide produced, they are sooner removed because the product removal is taken up by the Salmonella. They are taking the product NO2 inside. So they are dragging the amount of NO that is there, they are converted to NO2 and they are removing that product, so that the equilibrium shifts towards this left hand side, and there are very few nitric oxides left in the phagosome to create the damage on the Salmonella. And to uptake this nitric oxide inside, Salmonella creates a channel in their membrane, and this is called the nitrite transporter, produced by a gene called NIRC, and this transporter is also called NIRC transporter, nitrite transporter, through which they are taking it up. And as a result, nitric oxide no longer can create the damage. And not only that, whether it produces the SPIC proteins outside, this SPIC can also grow and it can stop and block the synthesis of nitric oxide in further level to regulate that no longer nitric oxide can be produced. And this SPIC can block the expression of the gene which is responsible for the nitric oxide synthesis. It is called INOS or nitric oxide synthase gene. So they are blocking this INOS gene, and thus by blocking INOS as well as removing nitrite, they are going to live there for longer time and going to reduce the amount of nitric oxide that is present inside the phagosome. Salmonella also expresses several enzymes that directly interact and inactive some reactive oxygen species we've already talked about, like superoxide dismutase, SOD, like other NADPH oxidases, like other molecules to block the NADPH oxidase, which usually generates H2O2. They can produce the hydrogen peroxidase to break down peroxide into water and oxygen. So using all of these different machineries, they can block the host or the phagosomal lysis machinery by the phagocytes. Now, adapting to the nutrient limitation encountered within the phagosome, that is very important, because inside the phagosome there is also nutrient limitation. Not only they need to survive against the phagosomal machineries, but also there will be limitation of nutrients. And how they are coping with that? They can cope up with that using the de novo synthesis genes that are present inside the Salmonella genome. So the de novo synthesis genes like essential metabolites, including aromatic amino acids and purines that are required, they can produce those purines and their necessary aromatic amino acids and some metabolites on their own inside those phagosomes, so that they don't need to rely on the host cell for that. So including all of these things are giving the Salmonella greater advantage during the survivability inside the phagosome for the longer run, or for causing the systemic enteric fever. And getting those important properties or resistance properties, or gaining importance pathogenicity island proteins, are giving them much more advantages for staying inside the host cell for longer period of time, and they mainly get all those important genes and genetic elements via horizontal gene transfer. And the loss of some functional modules is also there, gaining of some important elements also there, but ultimately is the blessing for Salmonella to stay there for longer period of time. And the most important part for getting those important mediators, important factors, important genes to survive there for longer period of time are mediated by integrons. Integrons are very new concept. They are one of the genetic elements involved in the adaptation of bacteria, and this is a very fast kind of adaptation, not very slow like evolution, very rapid kind of adaptation. And we'll address this involvement of integrons and how this integron involvement are taking the bacterial evolution so far. We'll be talking about it in the future discussion.